So I've drawn a nephron, but it's a little bit different than the one I showed before. This is not at all anatomically correct, but it's the best way to visually see what's going on. So as we've talked about, there's 4 different sections of a nephron. We've got your glomerulus and your Bowman's capsule, your proximal convoluted tube, your loop of Henle right here with the descending and ascending limbs, your distal convoluted tubes, and again distal referring to the fact that it's farther away from the Bowman's capsule, where the proximal is approximately right on top of it. Distal is away from it a further distance, and finally our collecting duct. Now we've got a blood vessel right here that would be coming off of the renal artery, and so here's our afferent portion, and then once it's mixed up in the glomerulus and the Bowman's capsule has surrounded it, then it's the efferent arteriole, which lends towards the peritubular capillaries. Now I'm showing this as one single vessel, but in fact we'd have vessels kind of contorting all the way around the proximal convoluted tubule, all throughout the loop of Henle, all along the distal convoluted tubule, and even running parallel to the collecting duct. And in this video, we're really showing how is blood moving, how is the filtrate moving, what's happening with different minerals, and at the end of this, you should be able to understand why it is that urine is able to become concentrated as it moves out the collecting duct, and exactly what the composition of urine is. So we'll go ahead and start by taking a closer look right here at the glomerulus. We're going to go way in to really understand what's going on. So a couple of things are happening here. A couple of things that you should notice. First of all, I've drawn it like a little ball, as if the blood is just coming in, moving through, and coming out. That's not at all correct. Remember from the previous video of our micrograph that these blood vessels are all mushed in there really, really tight. But the other thing you'll notice, and this was not a mistake, is that your afferent arteriole has this really wide diameter, whereas your efferent blood vessel arteriole has a really small diameter. And what this is going to force is a lot of pressure all throughout the glomerulus. This whole area is just full of high pressure blood. Not all the blood that comes in is going to be able to go out. And in fact, the blood vessels themselves can't get out. So all of the blood vessels are going back along this path here and continuing along. But a lot of the blood plasma is making it out. And we call this blood plasma a filtrate. And so there's a ton of blood plasma getting forced out. And there's a couple of ways that it's doing this. So I'm going to zoom in just a little bit more and take some notes in here that you should probably make mention of. You can annotate a diagram or write it on the side. The process that we're looking at here is called ultrafiltration. And it's caused because of the higher blood volume coming in than what can leave. So you have a high blood pressure within the glomerulus. And roughly 20% or so of plasma is going to be pushed from the capillaries into the Bowman's capsule, which you can kind of think as an open space that is just kind of circling around the glomerulus. So we've got this high blood pressure, and we also have what are called fenestrations. And fenestrations are very unique to the glomerulus in that the blood vessels have like a kind of slits all along them, which is going to allow and so those tiny little slits are going to stop any kind of red blood cells from making their way out. They stay in the capillaries while a lot of the liquid plasma gets moved along and then gets ultrafiltrated by what's called the basement membrane. And the basement membrane is only going to let some substances through. So that is going to make what is known as the filtrate. And the filtrate will contain water, glucose, amino acids, and tiny proteins, urea, and salts. And so here's our filtrate right here. Water, glucose, amino acids, urea, and salts are getting pushed into the Bowman's capsule. So now we'll kind of travel along here to the proximal convoluted tubule, and as a reminder, the chemicals that we have, or the substances that we have in the filtrate, are going to be glucose, amino acids, urea, salts, and those particular salts are going to include sodium, potassium, magnesium, calcium, and finally we have water. Lots and lots of water. And so we don't want to lose certain substances. If we lose them, then they're going out with the P, because if we kind of take a quick zoom out here, we remember that all of this is going to travel along all of the nephron until it makes it into the collecting duct, and then finally make it into the urinary bladder and be excreted. We don't want that. So we need to zoom back in here to the proximal convoluted tubule, and what's happening here is that we have pumping and passive transport of particular substances. Glucose and amino acids and small proteins are getting actively pumped back into the peritubular capillaries. That way we don't lose them, and close to 100% gets actively pumped back. Okay, urea is going to stay for the most part because that's what we're trying to get rid of. We've also got active pumping of sodium, potassium, magnesium, and calcium. Most of it, not all of it, but most of this gets pumped back. And because this is getting actively pumped back into the peritubular capillaries, water is going to follow because of osmotic pressure. Okay, so now we can kind of eliminate glucose and amino acid from our filtrate and move along to the next part, which is the loop of Henle. And so for the most part in the loop of Henle, what we're going to be trying to do is concentrate what we've got so that we're not excreting quite as much water. So our filtrate is going to be moving from the proximal convoluted tubule into the descending limb of the loop of Henle, but really where our story needs to start out in talking about the loop of Henle is the fact that in our ascending limb over here, we're losing or reclaiming a lot of the sodium and a lot of the chloride ions. I'm not really sure why my phone is doing this, but Anyway, so you're losing a lot of sodium and chloride ions out that goes into the tissue. And what that means is that in the descending limb, because there's a hypertonic solution out here, that we're going to start losing water. So water is going to be reclaimed back into the blood vessels. Okay, and so that's going to concentrate all of the filtrate as it moves into the descending limb of the loop of Henle. So down in this region, we're looking at very highly concentrated filtrate, very rich in urea, and that's what you want. You don't want to lose out all of your water, but you do want to try to get rid of as much urea as possible. And finally, we can slide back over here to the distal convoluted tubule. This is the last little bit of the puzzle before moving into our collecting ducts, because at this point we're going to also reclaim just a little bit more of that water in our salt. And so this is done by pumping in the salt back into the blood vessels from the nephron itself, and wherever sodium goes, water will follow. And again, my pen is not seeming to want to write very well right now, but that's okay. So sodium chloride is going to be pumped into the peritubular capillaries, and water will follow. So we've further concentrated our solute. That doesn't at all say what I want it to, but I think you get the gist. And finally, we're going to move here into the collecting ducts. And so at this point, we've got a concoction that is pretty much looking similar to urine. It's going to be high in... Here we go, sorry. Aha, I figured it out. Okay, so when we're talking about excretion, our filtrate at this point in the collecting duct is going to be very high in water and in salt. And additionally, a lot of the urea that we've been trying to get rid of is going to be gone as well. So that is going to go to the collecting duct, into the renal pelvis, into the ureter, into the bladder, and be excreted and out of your body, all for the purpose of trying to minimize how much urea we have, and as well as getting rid of excess water and salt if we're in a situation that we have a low solute concentration in the rest of the blood. So let's follow through and see if that makes any sense. So here's the nephron that we started with. Getting a little jumpy, sorry about that. Our afferent blood vessel, or arteriole here, is bringing blood into the glomerulus. A high concentration, I don't know why that's over there, but a high pressure is pushing a lot of the plasma into the proximal convoluted tubule via the Bowman's capsule. So you've got this high concentration of glucose and amino acids, urea, different salts, and water. The glucose and the amino acids, because of their high importance to the body, need to be reclaimed. So active transport is going to pump that and reclaim that back into the peritubular capillaries so that it can stay into the blood system and not be excreted as waste. Urea and salts are in here, and most of the salts, but definitely not all of them. Many of them are also pumped back into the peritubular capillaries. As we move down into the loop of Henle, we've got salts being reclaimed during in the ascending limb, and that's going to make a hypertonic solution relative to what is inside of the loop of Henle here at the descending limb, which is going to force water out. So the water is going to be forced out, reclaimed back by the blood system here. You've concentrated all of your filtrate. You do some fine tuning to determine exactly how much water and sodium chloride you want to reclaim. That's going to be based on the antidiuretic hormone. And then your filtrate is excreted, and most of that is salt, water, and urea.